When we left off Tuesday, we had talked about the ventricular system of the brain where CSF is produced. It's stored within the system until it leaves to encapsulate, to circulate around the brain, and it provides some buoyancy and protection for the brain and spinal cord, everything in the central nervous system. So what I want to start with is the route that it takes. We know that it surrounds the brain and the spinal cord, but it's produced in the ventricles. How does it get from one place to the next? You recall the choroid plexuses are what produce cerebrospinal fluid. So I'm going to do this first. I think the problem with this being so slow was that the videos that I'm recording end up being like 1.8 gigabytes, and it's really taxing for the system. So if I do this now, hopefully I'll get it out of the way and it'll still be quick. The 2D picture that you have shows you the lateral choroid plexuses, the choroid plexus in the third ventricle, but I find it a lot nicer to look at it here. The faded ventricles, the entire system are shown here with the cerebellum and the brainstem that are cut in half. We're looking at those in more detail today. And the choroid plexuses in the lateral ventricles are these things. So you can see those long tubes inside the ventricles. Those are the choroid plexuses that produce cerebrospinal fluid. And so it'll fill up the lateral ventricles. The only place for it to go when they're full is into the third ventricle that way. Did that work? That's pretty close. The third ventricle is this structure here. Oh, it's still slow. Which has its own choroid plexuses. You can see a very similar long tube going all the way down. That region, those structures, continuously produce cerebrospinal fluid. So it's a lot easier to get a sense of it from the model than I think from the 2D diagram here. Just clear that for next time. Because if I were to walk through this flow chart with you, which is the point A to point B flow of cerebrospinal fluid, we're going to start with the choroid plexuses in the lateral ventricles, highlighted here. Doesn't look anything like it did in the model. It fills up the ventricles and moves through what are called the interventricular foramina, or foramen. The really small, thin tubes that connect the lateral ventricles to the third ventricle. And a foramina is simply a fancy word for tube. Foramen is multiple fancy words for tubes, and you'll see those repeated throughout. We use foramen to address a number of different openings between different compartments. Those foramen connect to the third ventricle, where again we have more production of CSF. The third ventricle you can see in greater detail here. It's this entire open space, and the choroid plexus sits on the roof of that space. And we're following the purple arrows as we move through into the fourth ventricle, where we have another addition of cerebrospinal fluid. And this continues all the way down, but it's closed at the end. So how does it get out? How does the cerebrospinal fluid get out? It gets out right at the base of the fourth ventricle, through what are called the median and lateral apertures. And going back to this diagram, you can see them a bit better here. The small little arm on the left-hand side. That was good. We got this earlier than last time. See the small little arm. This is where we were. That small arm down here on the left-hand side pointing out. Okay. Maybe I'll take a screenshot for you and put it up online. No, all hope is lost. I'll take a screenshot, put it online, or I'll put it in a small video online, but just trust me, there are openings to the sides and openings to the back. You can follow the purple arrows here that then allow the cerebrospinal fluid to circulate around the brain, around the spinal cord. And that space all the way around those sensitive structures is one that we looked at last class called the subarachnoid space. Remember, I pointed that out specifically. It's a bit of a gap underneath the arachnoid layer of the arachnoid meninge. And the CSF will support and help protect the brain from impact shocks, etc. Now the CSF is constantly produced. So it's constantly moving through the ventricles, it's constantly being supplied and circulating around the brain. But where does it go? If it's constantly being produced, does it just fill up and fill up and fill up? Of course not. It needs to go somewhere. It's recycled, we get rid of it. And we get rid of it by dumping it into the venous blood that returns back to the heart. So cerebrospinal fluid, remember we talked about how it was produced, and it's filtered blood, essentially, circulates through and around the brain and is returned to the blood when it's done. It returns to the blood through the arachnoid villi. I made a point of identifying those to you last class. They were the things that sat in that venous structure at the top of the brain, and you can see one highlighted here, that protrusion that goes into the venous blood where CSF returns, mixes with blood, and then drains out of the brain, out of the head, and goes back to the heart. So we've completely followed this entire flowchart. From the subarachnoid space, drained through the arachnoid villi to the heart and lungs, and then blood returns to the choroid plexuses to make CSF anew. So that's the cycle of CSF flow within and around the brain. When we had the model, I hope that helped solidify the idea for you. Did that make sense, though? And you can follow this flow chart and identify each of the steps and where it might be and why it's important. Now we've connected to a concept that we aren't going to spend a lot of time on, at least not as much time as it should entail or is required. Blood flow to the brain, ultimately important. Without adequate blood flow to the brain, your body does everything it can to restore blood flow. And I'll talk about that in a second. The brain needs a constant blood supply, constant arterial blood supply. It's achieved through the carotid arteries mainly. There are also some other accessory arteries that I'm not going to talk about. But the main supply is through the carotid arteries in red, through all of the smaller arteries and structures of the brain, and then ultimately draining through the jugular veins. And this whole process is to support production of cerebrospinal fluid. It's to deliver glucose. The brain loves glucose. It has a really hard time using fat for energy. It removes CO2 and waste products. It delivers hormones. and it can transmit some substances that are produced in the brain as signals to other tissues. And as one example I've listed here, brain-derived neurotrophic factor. It's something like a hormone that the brain produces. It affects the shape and structure of neurons in the brain, but it also circulates in the blood. And we can measure it. If we take a blood sample, you can measure the production of substances like this at rest or during exercise. So this is really important, this concept. We need constant blood supply, constant oxygenated blood supply. As little as 4 minutes without blood supply can cause permanent brain damage. So we need constant, adequate, oxygenated blood supply to the brain. It's so important that we have a system set up in case one of the arms, one of the carotid arteries is ever blocked or insufficient. These are the carotid arteries, by the way. We're looking up towards the ventral surface of the brain. The carotid arteries are these openings here, so blood is flowing to the brain, mainly through those pathways, and then it diffuses as soon as it gets there through all of these other arteries. And one structure that you might notice right away, these arteries converge and form a loop. There are communicating arteries between the two carotid arteries that join together. It's a special structure called the circle of Willis. And its main job is redundancy. If your right carotid artery is somehow blocked or not delivering an adequate flow, the left carotid artery might be able to still supply all the structures of the brain. We have this redundancy enabled, where more blood flow from the left side can move towards the right if it's lacking on that side. So the circle of Willis creates redundancy that ensures there's always constant blood supply to the brain. Very important. In brief, those are the structures that I want you to know. What do I want you to know? Blood supply is important. What is supplied in the blood? How it gets there, carotid arteries. How it leaves, jugular veins. The circle of Willis creates redundancy. But this is an oversimplified, brief discussion of blood supply in the brain. It is much, much, much more complicated than that. And this is the actual routing of arteries through a person's face, skull, brain, head. The Body Worlds artists that create those structures will create these molds of the arterial branching within a body by pumping in this special latex material and then removing or dissolving everything else around it. And so what we're left with is an impression of all the fine branches on the arterial side where that latex was pumped, and everything else is removed. So there's a lot of opportunity for arterial supply to all the regions of the head. It's one of the most important places to deliver blood, period, in the body. Why is it important that the brain receives blood flow? Well, at rest, it uses 1/5 of the oxygen consumed to produce energy. Oxygen consumption is a measurement that we'll always make to figure out how hard your body's working. And sitting here at rest, well, you aren't at rest. You're thinking actively and you're taking notes. But at rest, your brain uses 1/5 of the oxygen your entire body uses. That can change with really hard thinking and note taking. It might go up to 25%. Not much more than that. Any interruption, though, on the order of 4 minutes or more, causes permanent damage. can weaken cells in the brain, causes death of cells in the brain, and then the associated functions that they provide are lost as well. So we always need oxygen because we're always making energy with that oxygen. The other ingredient that we need to make energy successfully is sugar, glucose. The brain doesn't store its own glucose. It's too busy. It has too many other things, synapses, pathways, white matter, gray matter, different regions. Signals are always being sent. It can't store its own glucose, so it needs a constant supply of not only oxygen, but glucose. And if either of those are lacking, the brain is smart enough to say, these shouldn't be lacking. Why are they lacking? Let me do whatever I can to restore flow to normal, and you end up horizontal on the floor, because a lot of the time, the problem with delivering blood to the brain is that it sits above the heart, and gravity's always pulling blood down. And if you are ever standing up vertically and somehow really relax, the blood can drain from your head and you will faint. We used to do this as an experiment in one of my undergrad laboratories. We had a special table with a plank on one side that you would attach a person to, and the plank could rotate. There's a hinge system on the table. And we got them to sit there on a little bit of an incline, and they would just relax. And you are essentially just supporting them and trying to drain all the blood out of their head. And without fail, you think you're not going to, but you will faint. And the table is set up that way because when you do faint, it's your job to watch them and then tilt them down to horizontal to make sure that the blood can flow back to the brain easily. You remove the gravitational column, which is really difficult in some cases for the heart to overcome and always ensure adequate supply of blood to the brain. We since lost, it has since become unethical to do that with undergrads. So we don't do that anymore. It's another reason why diabetics need to be very concerned. If there's ever low blood glucose, glucose situation, the brain can sense that, and if it's limited in how much glucose it can get, it will try to equalize the situation and allow as much blood flow with glucose as possible. It doesn't change the amount in the blood, but it still makes you pass out or faint in order to alleviate whatever it can. So constant blood supply to the brain, very, very important, but it's dangerous. We always want blood because it contains oxygen and it contains glucose. But it also contains hormones, drugs, CO2, alcohol, any other substance that's circulating in the blood will be delivered to the brain as well. And those harmful substances could damage the brain if it weren't for this layer of protection between the blood and the brain tissue. That layer is called the blood-brain barrier. Lining the brain tissue, intervening between the blood supply and the brain tissue are just a series of tight epithelial junctions. The entire surface is lined with these tight junctions that prevent passage of harmful substances into the brain tissue. Not all substances are stopped. Some substances can't move between cells because the tight junctions are there, but they can diffuse through a cell membrane. Oxygen and CO2 readily pass through. Caffeine, thank goodness, is one of those substances that can diffuse through the blood-brain barrier. Steroids that are nonpolar, they can easily slip through the lipid membrane, as does alcohol. So these are some candidates, some things that will pass through readily. Maybe you want them to, maybe you don't. It is really effective, though, in blocking most other substances. And in some cases, that might be a problem if you're trying to administer a therapeutic drug that you want to pass through, the blood-brain barrier is usually very selective. And if we're lucky, it will maintain its integrity throughout our life cycle, allowing only the substances that should get through to get through. If it's ever damaged, if that selectiveness is ever called into question, or if it's ever compromised, then it's very difficult to fix. That means that some of these substances, some of these normally restricted substances, can move through easily and damage brain tissue. So we want tight regulation of what the brain is given, of what is delivered. Lots of oxygen and glucose. We want to keep tabs on things like CO2, and sodium is a good one. We'll come back to that as we talk about some of the lower brain structures today. We don't really want alcohol to get through or steroids too much. We want tight regulation if we can. Any questions about these concepts as we move on? Anything not clear about the flow of cerebrospinal fluid, or the few main points about blood flow that we talked about? So far, so good. Okay. So we are going to unpack and break down some of those structures that I introduced at the start of the section. The brain as a whole, with our 4 main categories, we're going to start unpacking those categories and learning about what each of them does. So we're going to start inferiorly and move superiorly. The brainstem is the first stop on our venture. The brainstem is composed of 3 substructures. This is the part of the brain that links the higher order thinking brain with the spinal cord. It's the interface point between the brain and the spinal cord. And it is composed of the medulla oblongata, the pons, and the midbrain. And those are listed from most inferior to most superior. Medulla oblongata is at the bottom, pons in the middle, midbrain at the top. And I think I mistakenly referred to the diencephalon as the midbrain last class. I'm having a moment of like deja vu where I thought that. They're not the same. If I said that, I was wrong. They're not the same. So the brainstem is this structure here. It's a major site where relay signals, nerves, are sent out from the brain to the body. And it's a connection point between the central and peripheral nervous systems. A lot of the major nervous signals coming from the brain, coming from the cortex, leave in the brainstem and then travel out to specific regions of the body. Digestive system, the upper limb, some of the important organs, etc. So it's a main site of receiving sensory input from the periphery and then delivering motor output to important structures like the diaphragm for breathing and the heart for pumping blood as needed. So let's look at each of these briefly. And in the notes that you have online, there are extra slides with more detail. I am still giving you that detail, but I'm not going through all the slides. So you have them there for your information as we go through. So, most inferior portion of the brainstem, the medulla oblongata, is continuous with the spinal cord. It's hard to tell where one stops and the other begins. If we find that, though, and can take a slice through the brainstem, and then looking from the anterior portion looking backwards, so this is the front, the anterior portion of the medulla oblongata. This is the posterior portion at the back. You can see the choroid plexus and the 4th ventricle. We just talked about that today. We can see some interesting structures, and this is one spot that's involved in the autonomic regulation of ventilation and of heart rate. There seem to be a number of centers that are concerned with ventilation and heart rate. Probably because those are really important activities and we want some redundancy. But we have some indication that there are nuclei, there are nerves here that are involved in the autonomic control of ventilation and heart rate. There are respiratory medullary centers, some specific neurons in the medulla that only deal with triggering ventilation. And there are cardiovascular medullary centers as well. So there are different areas within the medulla that deal with each of those functions. I'm not going to go in depth of all of the different structures, but I want to point out a couple really interesting ones. One of the first things you're noticing on the ventral surface is that there are these ridges. These ridges are conduits of nerves as they make their way down from the brain to the rest of the body. And these are called pyramids. The pyramids are the primary way that conscious motor activity or signals are sent to the body. And what's interesting about how these motor signals travel from the brain to the rest of the body is that at the medulla, they cross over at this special point called the decussation of pyramids, or decussation of pyramids. They cross over, which is why anecdotally, if you're right-handed, your left brain is activated, or if you're left-handed, your right brain is activated. If you go to kick a soccer ball with your right leg, you're sending signals from the left side of your brain. The signals originate from one side and then cross over. So the contralateral side of the body is activated by these motor signals. And contralateral simply means opposite. The signal arises on the right side and it's executed on the left side because of this crossing over of motor pathways. What I also want to point out that we'll come back to later today are these enlarged sections just posterior to the pyramids. These are round enlargements, a little bit lateral, just posterior, but still in the anterior half of the medulla. And these are called the olives, the medullary olives. Maybe they look like olives. We can't see their role or see their connections, but these provide input to the cerebellum, which we'll learn is involved in the coordination of motor plans, making a plan to do a task, to play a sport, to jump, walk, run. And it's this link that helps them fine-tune those plans. When you learn a new skill, you're relaying information through the olives of the medulla to the cerebellum. So those are the main features that I want to point out here. We'll come back to those in a second. There's a lot more, as you can see, and there's some more information on the slides that you have, but the main highlights that I want to show you are here. Also, I forgot, these bundles of nerves are referred to in the body as a tract. There are ascending and descending tracts. And a tract simply means a conduit, a bundle of nerves with signals being sent in the same direction that have a similar function, a motor tract, a sensory tract. And we will come back to this when we talk about the spinal cord in the next section, but I want to introduce this definition to you now. If I say tract, I mean a conduit of nerves, a group of nerves moving in the same direction, having a similar function. So this is the most inferior aspect of the brainstem. It sits right below the pons. And the pons is also known as a bridge. The name means bridge. It's the middle feature of the brainstem, just superior to the medulla oblongata, and you can roughly approximate it as this highlighted green circle in the middle. The pons is a relay station. This is one of the main points where the two hemispheres of the brain connect, where the cortex will connect to the cerebellum, and where nerves pass through from the higher order regions of the brain through the spinal cord. This is a major relay station that links these signals in the body. It has a role in breathing. It helps the medulla regulate breathing by providing two really key aspects. And these are complicated terms, so I spelled them out for you. It smooths the breathing process. The main centers that control breathing were located in the medulla, but the pons has input into how those centers activate, how and when those centers activate. So one of the first areas that's immediately in contact with the medulla that helps with regulating breathing is the pneumotaxic area, which monitors how full the lungs get. Prevents overinflation. It ceases breathing, it ceases inspiration when the lungs become too full. And helps transition from a breath in to a breath out. The apneustic area times the breath. When you take a breath in, why do you immediately turn around and start to breathe out again? Well, the muscles want to recoil, but what signals you to exhale? It's the actions of the apneustic area that prevent these long pauses between breaths, that allow you to move smoothly from one in-breath to an out-breath and vice versa. Smooth integration and the repetition of breathing in and breathing out by these areas in the pons. I found this was also quite interesting. I don't know if you went home and watched that Mind Explained series of videos after I mentioned it last class, but the one on dreaming talks about how normally when you fall asleep, your brain shuts off initially. But when you enter REM sleep, you have all of these active signals as if you were perceiving the world around you awake. There are signals to activate your arms, to activate your heart and your lungs, to view different objects, to imagine different places. All of those signals occur within the brain as if you were awake. But the reason that you don't jump out of bed, the reason that you don't flail wildly, hopefully, is that the pons limits those signals. It stops the signals from reaching the rest of the body. As this major point of connectivity, as this bridge, the pons has a critical role in making sure that your dreams don't become a reality while you sleep. It limits the signals that are apparent while you dream. Pretty interesting stuff. So medulla, pons, we have a general understanding of what those do. The last section of the brainstem is the midbrain. It's the most superior section, and it's here that we have some connection to the cerebellum, which I'm not really playing up right now. What I want to show you here at the midbrain are these nodes of nerves that are involved in automatic responses. So the midbrain is located here under this green highlighted portion, and you can obviously see 4 round projections. Notice as well Notice as well, this is the opposite view. We've been looking at the brainstem from the front looking backwards. We've flipped that 180, and right now we're looking at it from behind. This is a posterior view. And so we've removed the cerebellum that would block our view if it were there, and we're looking at these 4 nodes that protrude from the back of the midbrain, called the colliculi. Collectively, the 4 of them together is called the tectum. You might also see it referred to, I think the book mentions this, it's an antiquated term called the corpora quadrigemini, which simply means body of 4 pieces, or 4 twins, body of 4 twins. And these are important relay points for warning signals below your conscious awareness, for automatic warning signals. They're grouped into 2 sets, superior and inferior colliculi. The superior colliculi are always processing visual stimuli, but they're not always active. They become active if those visual stimuli cross some threshold. If they are really close to you, if they're coming out of nowhere, if they're very quick, if they're abrupt, if you need an automatic response to them. And the inferior do something very similar with audio stimuli. They're always active, they're always monitoring, but they don't always relay signals unless there's a very loud bang, a gunshot, an explosion, cymbal crashing behind your head. And it's these relays that are responsible for the twitch, the urge to leap out of your seat, the automatic recoil if you hear something that's really close to your head, to move your head out of the way. Before you can even know the sound is occurring or that visual flash is occurring, these signals will relay through the colliculi, activate the muscles of the head and neck, activate the torso muscles to get you out of the way, whatever needs to happen in that startle response to get you out of the way. These structures are responsible for that startle response to audio and visual stimuli. There's one other structure that I want to point out to you in the midbrain, and we have to cut it open to see it. It's a pair of structures, but I'm calling it one. So if I cut the midbrain across and go down from the top, this is the anterior portion, that's the posterior, so the tectum with the colliculi is at the top of the slide. Those are the protrusions we just talked about. What I'm focusing on now are these obvious red regions, the substantia nigra and the associated red nuclei, which are here, these 2 paired together. They look like they're separate, but they're in close communication. And these are involved in coordinating smooth and unconscious movements. Let's say I'm at home making lunch and I want to open the cupboard and reach in for a can of soup. I don't have to plan about moving my arm up and not moving it down. I don't have to plan about where it's going side to side. I can just reach in and grab a can of soup. It's the actions of these 2 structures that allow that smooth, unconscious movement. We know this because when these structures are damaged, you lose that smooth, unconscious movement. The ability to make a plan to grab soup becomes your arm moving left and right, up and down. You're trying to reach for a can of soup, but you don't have that coordinated plan of action. And what I'm describing to you is Parkinson's disease. A characteristic of Parkinson's is damage to these structures. the loss of coordinated movements, the loss of that unconscious plan. So very important structures. We are behind. What isn't obvious from the slide that I showed you at the start, the brainstem, is its relation to the other structures around it. I want to introduce this series of structures now because it's a theme that we'll come back to throughout talking about these other structures. The reticular formation spans multiple areas of the brain. And it's pretty interesting because this is what will allow you to be aware of some signals returning from the body. Some people think this is part of the reason that you are conscious of sight, smell, touch, your position of limbs in space. I'm not going to say that this is where consciousness lives or the seat of your personality. I don't think anyone has that answer. But the reticular formation is the pathway that links sensory information coming back from the outside and brings it to your awareness. I'm aware of the sensation of touching the desk because the reticular formation is relaying that information to my cortex. I'm aware of the pressure in my feet because it's being relayed through the reticular formation. The reticular formation is this purplish tube. And then the network to which it connects is the reticular activating system. Those are the projections that go out. So the signals move through that tube into the projections, and it raises sensory information to your conscious awareness. You can think about pressure. You can think about heat. You can think about ventilation because of this relay. So it brings these sensations to your conscious awareness. When you are fully engaged and aroused, this is turned on maximally. You are in the zone, you're noticing everything. You're fully present and mindful. This is activated fully. When you sleep, inactivated completely. Right now, in lecture, somewhere between the two, maybe. Hopefully closer to the first one. So in sleeping, this turns off, and this is the target of melatonin. For those of you that have ever had trouble sleeping and you take melatonin, melatonin specifically turns down the reticular formation, turns down the network, and the sensory signals being brought to your awareness. It depresses this formation specifically to allow you to go to sleep.